Welcome to week 5 of Applied Immunology, where we will continue our coverage of the adaptive immune system by learning about B-cell biology. This is Lecture 5A, which kicks off the B-cell module for this course by discussing the process of B-cell development. We will briefly revisit the structure of immunoglobulin proteins today, but please refer back to our week 4 course materials related to the structure and generation of lymphocyte antigen receptors, just to make sure that you understand the basics of immunoglobulin molecules before you get started on week 5 materials. Today's lecture will primarily focus on the processes involved in B-cell development in both the bone marrow and spleen, including numerous B-cell selection checkpoints that guarantee the generation of a functional repertoire of B-cell clones. The remaining lectures this week will build off of this one by next exploring B-cell signaling downstream of BCR activation, as well as learning more about the functional contributions of B-cells and antibodies towards successful immune responses. Much like with our lecture on T-cell development, let's start with this differentiation plot on the left. Again, this shows how hematopoietic stem cells in the bone marrow can be differentiated into common lymphoid progenitors, or CLPs, which are the progenitor cell type for all lymphoid cell lineages that are listed at the bottom. Today we will focus on B cells, which again express a lymphocyte antigen receptor called immunoglobulin, which can exist in a membrane-bound form, where it is called the B cell receptor, or BCR, as well as the secreted form, where it is called an antibody. Immunoglobulin can bind directly to soluble or multivalent antigens, so it differs from the TCR in that it doesn't need to recognize foreign antigen on a self-MHC complex through MHC restriction. When the BCR binds to cognate antigen, B cells often proliferate and assume effector functions, which we will discuss later on this week. To quickly recap the structure of immunoglobulin, we've already learned that immunoglobulins are made up of heavy and light chains, and that both of these contribute variable CDRs that make up the variable region at each tip of the Y-shaped molecule. This variable region is responsible for binding to antigen, shown here as the little gray circle. Also remember that the light chain can come in two isotypes, kappa or lambda light chain. Similarly to our lecture on T-cell development, we'll start off today with the overall question of how exactly does a common lymphoid progenitor transition from a multipotent state to differentiate into a functional B cell. We know that a functional immunoglobulin must be able to bind to foreign antigen, though again this is different from the TCR in that foreign peptide recognition is not MHC restricted. We also know that, similarly to TCRs, immunoglobulin must remain ignorant or non-reactive to self-antigens that are expressed by healthy tissue, and this is in order to preserve immunological tolerance. With these two criteria established, let's next learn more about the development processes that yield functional B-cell clones from a common lymphoid progenitor. We can separate out the early stages of B-cell development which take place in the bone marrow versus later stages of development that take place in the spleen. In this diagram, we will work step by step through the main stages of B-cell development, which will increase in maturity as we move from the left to right across this slide. In addition to the organs where development takes place, each stage can also be separated according to whether development occurs in an antigen-independent or antigen-dependent fashion. We already know that B cells arrive from the common lymphoid progenitor, or CLP, so what happens next to convert a CLP into the first step towards a mature B cell? The first stage is something called an early pro-B cell. This is a lineage-defining stage where stem cells are committed towards B cell differentiation, and it is defined by the expression of the B cell-specific transcription factor E2A, which in turn induces expression of the early B cell factor, or EBF. These cells must also receive a pro-survival signal through the cytokine IL-7 that's present in the bone marrow in order to progress to the next stage of development. So these cells must also express a functional IL-7 receptor. Early pro-B cells generate late pro-B cells, which are defined by the rearrangement of V, D, and J regions on the immunoglobulin heavy chain. At this stage, late pro-B cells are subjected to the first checkpoint of B-cell development, which tests the functionality of the heavy chain in a complex called the pre-BCR. This process is analogous to beta selection in T-cell development, which tests the TCR beta chain in complex with the pre-TCR. Similarly to the pre-TCR, the pre-BCR's functionality is determined independently of any antigen binding to the pre-BCR. Pro-B cells that have assembled a functional heavy chain receive a survival signal through the pre-BCR, which allows them to progress to the next stage as pre-B cells. pre-B cells undergo recombination of their light chain, which allows them to assemble a candidate BCR consisting of their established heavy chain with a recombined kappa or lambda light chain. These candidate BCRs are expressed on the cell surface with a constant region isotype called IgM, and once the candidate BCR is expressed, the developing B cell is then classified as an immature B cell. The candidate BCR expressed by immature B cells in the bone marrow must be tested for autoreactivity to self-antigen, which represents the second checkpoint of B cell development. This checkpoint consists of the first round of negative selection, where B cells whose BCR induces strong signaling in response to binding self-antigens expressed in the bone marrow are not allowed to progress in development. Notably, from now on, all testing of the functionality of the BCR occurs in an antigen-dependent fashion. Autoreactive immature B cells can undergo 4 possible outcomes to either eliminate or inactivate autoreactive clones, which we will discuss in upcoming slides. Because these processes ensure the subtraction of autoreactive B cells in the bone marrow, which is a primary lymphoid organ, this checkpoint is responsible for establishing something called central tolerance. Immature B cells encoding non-autoreactive BCRs are allowed to leave the bone marrow and enter circulation, which next brings them to the spleen. These B cells are still immature, although once they've reached the spleen, they are referred to as transitional B cells. Transitional B cells undergo a second round of negative selection in the spleen, which tests whether the BCR recognizes any self-antigens that were not expressed in the bone marrow but may be found in other parts of the body. Transitional B cells that exhibit strong BCR signaling in response to self-antigens can be subjected to 2 possible outcomes, which remove them from the mature B cell pool, which we will discuss shortly. The elimination of these B cell clones establishes peripheral tolerance, or immunological tolerance of peripheral antigens. Only once transitional B cells pass this third checkpoint of peripheral negative selection are they then allowed to develop into mature B cells, which are characterized by the expression of their BCR on both IgD and IgM isotypes. The majority of these mature B cells reside in the spleen or lymph nodes, where they receive survival signals and await activation through the binding of cognate antigen to undergo activation, which is what we will cover in our next lecture for week 5. Like with T cells, it can be helpful to view how these developmental stages are defined according to the recombination and assembly of antigen receptor genes through either VJ or VDJ recombination. As with T cells, B cell development typically follows a pattern where successful gene rearrangement events yield proteins that function by relaying a survival signal to the developing B cell, and this is what allows it to progress to the next stage. The early stages of B cell development occur in the bone marrow and correspond with VDJ recombination events at the immunoglobulin heavy chain locus. First, D and J segments are joined at the early pro-B cell stage during B cell lineage commitment. This is followed by V joining to the recombined DJ segment at the late pro-B cell stage. The first pre-B cell stage, called large pre-B cells, is defined through a fully recombined immunoglobulin heavy chain consisting of recombined V, D, and J segments, which then must be tested for functionality. To do this, the heavy chain is paired with one of 2 possible surrogate light chains, lambda 5 or V pre-B, which form an immunoglobulin-like molecule called the pre-BCR. Functional heavy chain proteins can signal in an antigen-independent manner through the pre-BCR, which triggers survival signals that allow the pre-B cell to proliferate and advance to the next stage of development called small pre-B cell, which is where light chain rearrangement occurs. Note that at this stage, the heavy chain has been recombined with the constant region labeled with the Greek letter mu, which means that the pre-BCR is expressed transiently as an IgM isotype. Successful signaling through the pre-BCR also prevents further recombination at the other heavy chain alleles in the cell, which enforces the concept of allelic exclusion, where only one allele of the heavy chain can be expressed at a time. There are many parallels here to the beta selection checkpoint in developing T cells. The surrogate light chains for the pre-BCR are analogous to the surrogate alpha chain that forms the pre-TCR, and the concept of allelic exclusion is enforced for both immunoglobulin heavy chains and TCR beta chains. Lastly, both pre-BCR and pre-TCR relay antigen-independent survival signals that induce the proliferation of B cells or T cells expressing functional heavy chain or TCR beta chain, respectively. The small pre-B cell stage is triggered following pro-survival signaling through the pre-BCR, and this is the stage where VJ recombination occurs at the immunoglobulin light chain locus. Like with the immunoglobulin heavy chain, the light chain also exhibits allelic exclusion as well as isotypic exclusion, which means that each immature B cell will undergo recombination at only one light chain allele at a time, and that only one isotype of light chain, either kappa or lambda, will be expressed at a time. Once a candidate light chain is rearranged, it is expressed by immature B cells in the bone marrow and must be tested for autoreactivity to self-antigen. This represents the second checkpoint of B cell development, which is a first round of negative selection against BCRs which induce strong signaling in response to the binding of self-antigens that are expressed in the bone marrow. Again, this selection occurs in an antigen-dependent manner. We will discuss the different mechanisms that are used to ensure the removal or inactivation of autoreactive B cell clones in the next slide, but I want to first reiterate the point that this first round of negative selection takes place in the bone marrow and is therefore responsible for establishing central tolerance. Immature B cells can be subjected to 5 possible outcomes following the process of negative selection. The first is that the BCR is non-reactive to any self-antigens expressed in the bone marrow, in which case the immature B cell can exit the bone marrow and travel to the spleen, where it can undergo additional steps towards B cell maturation. Note that unlike T cells that must recognize self-MHC, because B cells can bind directly to cognate peptides, they do not undergo positive selection, at least not to the same extent that MHC restriction must be confirmed through positive selection of thymocytes during T cell development. There are 4 outcomes that are reserved for autoreactive B cell clones in the bone marrow. So the first 2 options occur in a situation where the BCR recognizes something called a multivalent self-molecule. These are typically ubiquitously expressed self-antigens that are found in high abundance in a concentrated area. Because there are multiple antigen binding sites on the same molecule, that is, there are multiple valencies expressed on the same molecule, multiple BCRs on the same B cell are recruited to the same area where they can cross-link and signal strongly through their BCRs as a result. In this case of recognizing self-antigen on multivalent self-molecules, the B cell can undergo one of 2 fates. The first is simply to be eliminated through programmed apoptotic cell death, in a process we've already heard about, which is clonal deletion. The second possibility for immature B cell clones that recognize multivalent self-antigens is to undergo another round of light chain rearrangement. This gives the B cell another chance to generate a new light chain that can pair with the clone's existing heavy chain, therefore creating a new BCR with new antigen specificity. If this new receptor is still self-reactive to multivalent antigen, the B cell will either undergo additional rounds of light chain rearrangement or die through clonal deletion. However, if the new BCR is now ignorant of self-antigens, the immature B cell passes this first negative selection checkpoint and can then migrate to the spleen for additional maturation. This process of additional light chain rearrangements that rescue a B cell clone from clonal deletion is something called receptor editing. Beyond clonal deletion and receptor editing, autoreactive B cell clones can be subjected to 2 additional mechanisms that functionally inactivate them, and both of these are dependent on the types of antigens that they bind. Both of these situations differ from the clonal deletion and receptor editing outcomes in that they are induced following binding to soluble self-antigens rather than large multivalent self-molecules. The first option is shown in the 3rd column, where the soluble self-antigen has multiple antigen recognition sites that can induce BCR cross-linking. Cross-linking simply means that multiple BCRs can bind to the same single molecule of soluble antigen, which brings the individual BCRs close together and influences their downstream signaling, which we'll learn more about in the next lecture. Since these soluble antigens are smaller, they don't induce the same degree of cross-linking as multivalent molecules that we already discussed. So in this case, the B cells undergo a state of inactivation called Anergy, where they are rendered unresponsive to cognate antigen. These cells still migrate to the periphery and complete B cell maturation, though they predominantly express their BCR as an IgD isotype instead of IgM. And although these B cells are not removed during negative selection in the bone marrow, they usually fail to compete with competent B cell clones for survival signals in secondary lymphoid organs, so anergic cells usually end up dying within a few days. The second option is shown in the 4th column, where the soluble self-antigen is either monovalent or binds with low affinity, which means that no cross-linking occurs and BCR signaling is not engaged. Although these immature B cell clones could potentially be autoreactive due to their specificity, if the self-antigen is not sufficient to induce BCR signaling during central tolerance induction, then these cells are said to be clonally ignorant or immunologically ignorant. And this just means that even if self-antigen is encountered, these B-cell clones are harmless since they aren't actually activated in response to antigen. These B cells therefore undergo the same maturation steps as the truly non-autoreactive clones represented in the first column of this figure, so they actually successfully exit the bone marrow and migrate to the spleen to complete B cell maturation. Immature B cells that pass the central tolerance checkpoint in the bone marrow are then allowed to migrate to the spleen, where they concentrate in areas called B cell follicles. Once they arrive, these cells are referred to as transitional B cells, and they are subjected to the third and final checkpoint of B cell development before being allowed to progress as fully mature B cells. This last checkpoint is the second round of negative selection that this time takes place in the spleen. The negative selection of transitional B cells tests whether B cell clones with central tolerance are also non-autoreactive to additional self-antigens that are present in the periphery but not the bone marrow. This includes molecules expressed by splenocytes or other immune and stromal cells in the spleen, as well as soluble molecules which circulate in the blood. Because this last checkpoint tests for autoreactivity against antigens encountered in the periphery, this checkpoint again establishes peripheral tolerance of B cells. Transitional B cells subjected to negative selection in the spleen have 4 different fates, each of which we've already covered during the negative selection of immature B cells in the bone marrow. B cell clones that recognize soluble self-molecules, shown here in the second column, assume an anergic state. And again, because these cells cannot compete for survival signals in the spleen during the remainder of B cell maturation, they are eventually eliminated from the B cell repertoire by apoptosis. B cell clones that recognize monovalent or low affinity self-antigens assume a state of clonal ignorance, shown here in the third column, much like we just discussed during negative selection in the bone marrow. These cells, again, are technically autoreactive, but will likely never undergo activation upon binding to self-antigen, so they are viewed as functionally harmless, and therefore they're allowed to progress through the final stages of B cell maturation. Notably, B cells that recognize multivalent self-molecules in the spleen can only undergo clonal deletion through apoptosis, which is shown in the first column here. This is a key difference compared to negative selection of immature B cells in the bone marrow, which also have the option to undergo additional light chain rearrangements through the process of receptor editing. Transitional B cells are thought to be in a slightly more mature state than their bone marrow counterparts, where they are no longer able to rearrange genes at their immunoglobulin light chain loci, and this explains why receptor editing is not an option for saving these autoreactive B cell clones in the spleen. Lastly, transitional B cell clones that do not bind to any self-antigens in the spleen represent clones that exhibit both central and peripheral tolerance. These clones are the ones that will make up the functional pool of mature B cells that can be used to recognize and respond to foreign antigens. We will end this lecture with an overview of how transitional B cells complete their development in order to form mature naive B cells. As we just discussed in the last slide, transitional B cells that are unresponsive to self-antigens survive the negative selection checkpoint, which establishes peripheral tolerance in the spleen. This takes place in an area of the spleen called the B cell follicle, shown in these diagrams as a darker yellow region. The B cell follicle obviously has a high density of B cells, as well as specialized dendritic cells called follicular dendritic cells, or FDCs. Importantly, FDCs produce a ligand called BAF, which is an important survival signal for transitional B cells that is required for their successful maturation. Transitional B cells actually consist of 2 distinct developmental stages, which are called T1 and T2. T1 B cells are the immature B cells that just arrived from the bone marrow, which express their IgM isotype BCR as well as the receptor for BAF. BAF expressed by FDCs is detected on T1 B cells, which in combination with very weak stimulation of the BCR provides a critical survival signal that allows B cells to move to the T2 stage of development. Now, T2 B cells are defined by their upregulated expression of a B cell co-receptor called CD21. T2 B cells maintain BAF receptor expression and require another survival signal through BAF in order to progress to the final stage of B cell maturation. So, mature B cells are characterized by co-expression of both IgM and IgD isotypes of their B cell receptor, as well as surface expression of CD21 and the BAF receptor. Mature B cells can be split into 2 different subsets, follicular B cells and marginal zone B cells, or MZ B cells. The majority of T2 B cells that receive survival signals will become follicular B cells, and these are B cells that recirculate between the blood and B cell follicles of secondary lymphoid organs, including the spleen as well as the lymph nodes. A small proportion of T2 B cells that survive selection will become marginal zone, or MZ B cells, which occupy the intermediate region between white pulp and red pulp in the spleen, and also express high amounts of the coreceptor CD21. Importantly, due to their proximity to the vascularized red pulp of the spleen, marginal zone B cells are optimally positioned to be among the first immune cells to detect and respond to antigens and pathogens that circulate in the blood. Both follicular and marginal zone B cells are considered mature B cell subsets that are favored for precious space and resources in the B cell follicle. So they often outcompete immature B cells that are newly arrived from the bone marrow for this niche of B cell survival and maturation. We will learn more about the signaling, activation, and function of mature naive B cells in the upcoming lectures for our Week 5 B cell module in this course. To summarize, in this lecture we've learned that progenitor cells undergoing B cell development in the bone marrow and spleen must survive 3 major developmental checkpoints. These checkpoints exist to ensure the main criteria of functional B cells, which is that they must not react to self-antigen in order to preserve immunological tolerance, but they must be able to recognize foreign antigen. Only the B cells that survive through these 3 checkpoints can then go on to form mature naive B cells, which are further subdivided into marginal zone or follicular B cells based on their location in secondary lymphoid organs. We've learned that the testing of the pre-BCR and the 2 different stages of negative selection all differ with respect to which B cell development stage they act on, the type of testing that they exert on candidate B cell clones, their dependence on antigen, and the categories of immunological tolerance that they are responsible for establishing. You've seen this information a few times now throughout this talk, but some key facts are written out as bullet points on this summary slide. That's all for today's lecture, but please remember to watch the remaining 2 lectures for Week 5, in which we will learn more about the signaling pathways activated downstream of mature naive BCR activation, as well as the different effector functions of B cells and their secreted antibodies.